Good evening, everyone. And my name is Louise Warnig, and it is my great pleasure and honor to welcome all of you to the inaugural lecture of Professor Bruce Anderson in my capacity as Dean of the Faculty of Science. So, an inaugural lecture is a special occasion for our faculty and a time for celebration, and we are honored that you are here tonight to celebrate this special milestone in the academic career of Professor Anderson with us. Some of you here in person and some of you online. I would also like to extend a special word of welcome to Professor Teresh. Oh, please, yes, please sit down. My apologies. I would also like to extend a special word of welcome to Professor Daresh Ramjugarnath, the Deputy Vice Chancellor Learning and Teaching, who is representing the senior management of the university this evening, as well as Professor Ingrid Revitsky, our Vice Dean Learning and Teaching, and Professor Frikkie Scholes, our Vice Dean Research and Postgraduate Students. Also, Professor Teresa Vossler, the Executive Head of the Department of Botany and Zoology. A warm welcome also to Bruce's partner Lee and all family, friends and colleagues from Stellenbosch University as well as other universities, as well as current and former students of Bruce. A warm welcome to all of you. It is now my honor to introduce Professor Bruce Anderson. Bruce obtained a BSc Honour and a PhD in Botany from the University of Cape Town. On completion of his PhD in 2003, he joined the University of KwaZulu-Natal as a postdoctoral fellow, specializing in pollination. In 2006, he was appointed as a lecturer in the Department of Botany and Zoology at Stellenbosch University, and he was promoted to professor in 2020. Bruce currently has a B2 NRF rating and serves on the editorial boards of 3 international journals. Since arriving at Stellenbosch University many years ago, he has taught and coordinated a number of modules spanning all 3 years of the program in biodiversity and ecology, and often receives excellent feedback from students. As a toddler, Bruce aspired to become an entomologist because he was fascinated by creepy-crawly creatures. However, as a student at UCT, he discovered that plants are fascinating too, and it was specifically the interaction with insects, birds, and other animals that interested him. Its research focuses on the evolutionary ecology of plant-animal interactions, including pollination biology, herbivory, and seed dispersal. To quote from his bio-sketch prepared for this evening's lecture, Bruce is particularly interested in how interactions between different species can shape their evolution and behavior, despite the fact that interacting individuals are seldom invested in each other's mutual success. He marvels at the extraordinary diversity of life, continuously radiating as a product of entangled interactions within and between species, unquote. Recently, his research on using quantum dots or fluorescent nanoparticles to label and trace pollen has received much interest locally and internationally. Bruce, we are looking forward to hear more about your exciting journey with floral stowaways and hitchhikers that use animal riders to embark on journeys that often take them to unintended places. Friends and colleagues, Professor Bruce Anderson, please give him a round of applause.
Right, thank you very much, Louise, for the introduction. I'd also like to start just by thanking all of my friends and colleagues for being here today. It's really fantastic to see so many friendly faces in an audience. I'd also like to thank those who've logged in to watch this. And I'd like to give a special little hat tip just to my past students who are with me here today, who I can now consider to be my friends and colleagues, which is amazing. But what's really amazing is that I know that some of you were there on the very first lectures that I ever gave at Stellenbosch in 2006. So what an incredible journey that's been. Thanks for being here tonight. And to many of my friends sitting here, some of you have an inkling of what I do, but probably no more than that. And I know that sometimes we sit at a dinner table, we have supper together, we talk a little bit about work, and you may look across and see me drifting off, gazing into some distant corner of the galaxy, and you may think, geez, what's this guy got between his ears? And so I have a little confession to make, and that is that during those moments, there's a high likelihood that I'm thinking about sex. Plant sex. And you may wonder, how is it that plant sex manages to keep this guy's mind occupied for such long periods? And so today, while I have you as a captive audience, I would like to tell you a little bit more about what I do, and particularly why I find the sex lives of plants so interesting. For a start, Oh yeah, thanks, Don. I think that's as dark as I'm allowed to make it. For a start, plants, the sex lives of plants are often way more intricate and complex than the sex lives of mammals. And for a start, plants do some pretty crazy things. They routinely encourage the participation of other animal species in the most intimate parts of their sex lives, and they pay for those services using pollen and nectar as rewards. The other thing that plants do is that they like to mess around with gender roles. So I'm going to take you back to your boring old high school lectures now, and I'm going to show you a picture of a flower. And maybe you'll remember this little yellow thing here, that's the anther, the male part. And this pink thing here, that's the female part of the flower, it's the stigma. And what you'll suddenly realize is that flowers, the things that we call flowers, are really just collections of reproductive parts cradled in some beautiful petals. The other thing you'll notice is that these flowers are hermaphrodite. So they've got both male and female parts. And not all flowers are hermaphrodite. Some flowers can be male or female. Some whole plants can be either male or female. Some plants can be a little bit male and a lot female, or a lot female and just a little bit male. And they manage to traverse this amazing spectrum of gender strategies. And so it makes plants a really fascinating thing to work with. Now, the interesting thing about this particular picture here is that you can see the male parts of the plant are placing pollen onto the female parts of the plant. So the plant is effectively mating with itself. And while that can be a very convenient strategy at times, it's never a great idea to make a habit of mating with yourself, because it leads to inbreeding depression, right? So this is something that Darwin noticed, and he noticed that if you moved pollen between flowers on the same plant, the plants produced fewer ovules and fewer seeds, and often the offspring that they produced were of lower quality than if he had moved the pollen between different plants. And he concluded from that nature abhors perpetual self-fertilization. And ironically, it was this work that made Darwin a little bit worried about his own marriage, because he had married his first cousin. And he sometimes expressed worries that the health or the poor health of some of his children were the result of his first cousin marriage. Darwin also realized that many plants go to extreme measures to try and avoid self-fertilization. And he wrote an entire book about this, on floral polymorphisms, which he believed had evolved to reduce or prevent self-fertilization. And so this is actually a little picture from his book. So what's a floral polymorphism? Well, it's when you find a species which has two different kinds of flower in the same population, for example. And so in this particular example here, he looked at these primula flowers, and I don't think you can see it, but he's drawn a picture where these primula flowers have reproductive parts which are at different heights. And he hypothesized that was to encourage pollen movement between these different morphs rather than within. And I'll talk a little bit more about these polymorphisms later and give you a nice visual example. But one of the things that Darwin said about this research was, I do not think that anything in my scientific life has given me so much satisfaction as making out the meaning of the structure of these plants. Now, these are big words coming from the man who invented or made evolution fly. But some years after he wrote this book, he found out about a new polymorphism which he had never seen before. And it was described by a guy called Todd, and he was really excited about this polymorphism because Todd had described that certain flowers can be handed, left or right-handed. And so here's an example of this. This is the flower that it was originally described in, Solanum rostratum. It's related to things like potatoes and tomatoes in the same genus. But what you can see here is the female part, the stigma of this flower, is on the flower's left-hand side, whereas the anther, the main pollinating anther, is on the flower's right-hand side. So this is what we would call a left-handed flower, and this is what a right-handed flower would look like, with the female part on the right and the male part on the left. I've actually just mirror image switched these, so I've taken a shortcut. Those are not two separate flowers, but that's how it would normally look. Darwin was quite excited about this, but he was really sick at the time, and he wrote to Todd and he said, send me some seeds so that I can have the pleasure of seeing the flowers and experimenting on them. And this was to be his very last bit of scientific correspondence, because 9 days later he died on the 19th of April in 1882. Nevertheless, I'm fairly certain that Darwin probably had an idea of what handedness was about in plants. And I'd like to give you it's really just a caricature of how biologists think that handedness works. I'd like you to imagine a flower like this one, so a left-handed flower, and a whole plant consisting just of left-handed flowers. And now I'd like you to imagine a pollinator visiting this flower. Remember, the anther is on the right-hand side, so when the flower places pollen on the insect, it's going to be on one side of the insect's body. The stigma, however, is on the wrong side of the body to pick up that pollen. So any flowers that look like that on the same plant that the insect visits, it's not going to transfer pollen. But in this population, there will be certain flowers or certain plants which have the opposite configuration. And now, when this insect visits, the pollen is on the right side or the correct side of the insect to get picked up by the stigma. And the anther on this side will place pollen on the opposite side of the pollinator, where it can then mate with those plants. So you can see that if entire plants are either left or right-handed, it encourages this movement of pollen between morphs rather than within morphs, and this reduces the probabilities of self-pollination. So now, this is all in theory because actually no one's really been able to follow pollen grains. And so I'm going to tell you a little bit about some of the work that we've done in our lab on this plant over here called Wachendorfia paniculata, which you may find if you walk up the mountain tomorrow. They're busy flowering. And they can be left or right-handed, so entire plants left or right-handed. This is what a left-handed flower looks like. It's got the stigma, the female part, on the left, on the flower's left, and the right-handed plant has it on the other way. And they have 3 anthers, one on the same side as the stigma, and 2 on the opposite side. So now, I want you to imagine a pollinator visiting one of these flowers, and remember it's got 3 anthers. So 2 of the anthers are going to place pollen on that side of the pollinator, and 1 of the anthers is going to place pollen on that side of the pollinator. Now, if this insect visits another left-handed flower, you can imagine that 33% of the pollen placed, or 33.3, will be available to be placed on other left-handed flowers. Okay? The remaining 66.6, that should be available for transfer to right-handed flowers. So we hypothesized that, well, maybe this handed configuration will lead to pollen movement where 66% of pollen movement is between morphs and the remainder, 33, is within morph. But that was all in theory. We didn't really have a way to track these pollen grains because nothing seemed to stick to pollen grains. Regular dyes didn't stick. Fluorescent dyes didn't stick. And many people had tried in the past. And then I had a really wonderful student, Cornel Minnar, who worked with me, and he realized that, hang on a second, we can use fluorescent nanoparticles to mark these pollen grains. So these are semiconductor crystals. They're tiny. They're about the size of a virus, and they can glow. They glow with fluorescence if you shine an ultraviolet light on them, because the electrons start jumping around, and as they do so, they emit photons of light, and the wavelength of the light emitted is dependent on the size of those crystals. And so if you can constrain the size very precisely, you can control the amount or the wavelength of light being emitted. And so these nanocrystals are used, actually they've been used for decades now in industry, and many of your cell phone screens and your TV screens will use this technology. But I think this was probably the first time the technology was ever used in an evolution or ecological way. So using a pipette, now this is real bucket and spade science, using a pipette with some quantum dots in a solution of hexane, you could pipette this solution onto the anthers of flowers. The hexane would evaporate really, really quickly and leave the quantum dots behind. And the quantum dots have ligands on them which can attach to various substrates. And you can buy quantum dots which will attach to the lipids on pollen grains. And so we could use these quantum dots to label pollen on individual flowers. So if we found that pollen anywhere else, we'd know exactly which flower it came from. We could even distinguish pollen grains from different anthers in the same flower, which you couldn't even do using molecular techniques. We could then go and capture pollinators in this population, and we could look at those pollinators and see, oh, I know exactly where this pollen grain came from. In fact, I know which anther that pollen grain came from. And you can see a photograph here of one of these carpenter bees which we captured, and it's got pollen grains fluorescing in green and red, depending on which anther, on which flower they came from. We could even then go into the field, and after a bout of pollination, we could harvest the female parts of flowers, the stigmas, and we could put those stigmas under a microscope, and we could look at them and look at the pollen grains on those stigmas, and we could identify which male parts they came from. So now I'm going to dive into some results that we got using these ideas. And I'm very quickly going to show you what we found for these left-handed flowers. So I'm only going to focus in on the left-handed morph, because whatever happens to the left-handed morph also happens to the right-handed morph. It's just a mirror image flip. So we're going to look at these left-handed flowers, and we are going to ask a simple question. When you look at the stigma of a left-handed flower, so that's the female part, where does the pollen on that stigma come from? So here's a little, it's really just a pollen budget if you want, showing where these left-handed flowers get their pollen from. And I'm just showing you that they get about 15% of their pollen from other left-handed flowers. And these are the contributions by each anther. This anther here is the major left-handed contributor, and 12.8% of the pollen found on the stigma is from that anther, which is on the same side as the stigma. The majority of the pollen received by these flowers are from right-handed flowers, okay? In fact, they're getting about 85% of their pollen from right-handed flowers. So this is a lot higher than we had predicted. We predicted only 66%. So these flowers are doing a fantastic job of moving pollen between morphs rather than within morphs. And there are reasons for it, but I'm not going to go into it now. It's a little bit short for time. But I want to just highlight something that I think is even more interesting than the fact that Darwin was right again. And that is that almost all of the pollen found in the stigma is actually just from one anther, this green anther here. But there are 6 anthers in this system. This flower has 3 of them, and this flower has 3 of them. So these 2 bottom anthers seem to be doing nothing. This anther is doing very little. This anther is doing very little. This one looks like it's doing very little, but because it's a mirror image switch, remember, it's donating probably about 76% of the pollen to that anther. So actually, there are 2 anthers which seem to be doing a lot here. So what's the point then of evolving these anthers at the bottom? And what I think is happening, and we've started doing some work on this, is that we think that these anthers are maybe used in different ecological scenarios. And we think that maybe these anthers at the top, which are much more widely spaced, are useful for large pollinators like carpenter bees. And maybe these bottom anthers are useful for much smaller pollinators like honeybees. The other thing you'll notice here is that this anther actually is doing something. And so we think that when these plants find themselves in populations which are skewed towards one morph, in other words, if you found a population which was mostly this morph, there won't be a mate shortage for these plants, and they can effectively still mate with some individuals even though they are the same morph. So what's interesting about this is that you have these different anthers which appear to have evolved for different ecological functions, and they have different sexual functions, really. And if I had to draw an analogy with some kind of animal, you would have to imagine an animal with 3 penises, right? All used for different kinds of matings, maybe one for fun, and maybe one to mate with a regular sexual partner and make babies, and a third one, well, I'll leave that to your imagination. What I've tried to show you till this point is this very simplistic view of a pollen journey, the journey that pollen grains take from the anthers of a flower, onto the pollinators, and then finally onto the stigmas of flowers. And I've also tried to show you that using these amazing quantum dot tools, we can actually start documenting that journey, visualizing it, quantifying it, and distinguishing different kinds of pathways of these pollen grains. And before quantum dots came along, it was really difficult to do this because you would look at an insect, it would have pollen on it, all the pollen would look identical, and you wouldn't know where one pollen grain came from versus another pollen grain. But using this, we can actually start doing so. But just having this as a tool and a technique sort of changed my view of plants, and it made me start thinking of plants in a different way and asking questions which I'd never asked before and which I don't think many people had ever asked before. And so, what I'd like to do now is I'm going to move on to the next part of my talk. And I'd like to take you on a little journey, a journey where fact meets fiction and a little bit of fanciful imagination. And I'm going to allow you into my head a little bit. And I'm going to show you or give you a glimpse of some of the things that I see when I gaze out into space. And I've talked a lot about pollen movement until this point, and pollen movement sounds like a really, really simple thing. But actually, if you're a plant, the movement of pollen is quite difficult because you're rooted to the ground, and you've got to use these other animals to help you move your pollen grains around. And so this is the journey that I'm going to take you on. I'm going to take you on one of these pollen grain journeys, and we can basically break up this journey into 3 phases. The first phase, we can ask, what happens to pollen grains that are on a flower? Do they even actually start this journey? And actually, a lot of pollen grains don't start this journey. A lot of flowers bloom, and the pollen sits on the flower, and nothing ever visits it. The flower dies, and the pollen falls to the ground. Sometimes pollinators do arrive, but before they even get there, the pollen might be blown off into the wind and onto the ground. Or the pollinator arrives and, it bumps the pollen off the flower, and that pollen grain never really starts the journey. Or the morphology of the pollinator and the plant don't quite fit together, and the pollen never gets onto the pollinator. So there are a lot of interesting fates that await these pollen grains. But some grains do manage to reach the pollinator, and this is the second phase of the journey. What happens to pollen grains once they're there? And what is interesting is that once they're there, it doesn't guarantee you success. Some grains are successful and some aren't. So what is it about those successful grains which allow them to go on to fertilize ovules versus the unsuccessful grains? The last part of this journey involves what happens when pollen grains reach another flower. And let's say they do reach another flower of the same species, but a different individual. Well, the race hasn't quite stopped yet. These pollen grains now have to build a tube from the stigma of this flower down the style to reach the ovules. And then they send their little sperm cells down that tube to fertilize that ovule. It's kind of similar to the sperm races you may see in mammals, where sperms race to get to an ovule, although usually these will, the races between pollen grains will be from pollen grains of different flowers. So only the fastest pollen grains will get to fertilize those ovules. So what I've just described to you now is essentially an obstacle course for pollen grains, right? There are a whole lot of hurdles that pollen grains have to overcome in order to make it to their final destination. And each of these hurdles represents a wonderful opportunity for natural selection to select on floral form and pollen morphology, to select for more and more efficient pathways. And what's interesting is that there are so many different horrendous fates that await these pollen grains that I would consider this journey taken by these pollen grains as probably the most arduous and difficult journeys taken by the gametes of any species. And part of the reason for that is that plants have had to use this extra cog in the system, which no other animals really have to use when they mate. They have to use these pollinators. And what goes on here on the pollinator is a major source of pollen loss. And interestingly, this part of the journey here, the journey that takes place on the pollinator, is probably the least known part of this journey. And it's something that we've only really started to contemplate recently, and I'd like to do just that in the rest of the talk. I'm going to focus on what happens to pollen grains when they're on pollinators. In this particular picture, you can see the marvelous precision of pollen placement onto this pollinator's beak. And you might ask why it's so precise, why is there not pollen all over this pollinator? Well, if the pollen was anywhere else on this bird, it probably wouldn't be a very good place to be picked up by the stigmas of other flowers. So there's a lot of competition to get to that little sweet spot there. And sometimes, when I think of pollinators and when I stare out into space, I imagine these pollinators as buses carrying little pollen passengers, okay? These little green things here, these little green things are pollen grains. They're not COVID viruses. They're actually hibiscus pollen. But you can see a pollen grain bus which has just arrived here. And what's interesting about these pollen grains is that they are not the friends of the pollen grains residing in that flower. Those pollen grains are mostly from rival males, right? So if these guys go on to fertilize ovules and not your own pollen, you're likely to be relegated to the evolutionary dustbin. So it's imperative that when these buses arrive, and I must mention that these buses are not reliable, you don't know when they're going to come or if they're going to come at all. When these buses do arrive, these plants have to get their pollen onto them. And the question is, how do they do it? Because in most situations, all the seats are taken. And so there's immense competition to get those pollen grains right there and to occupy the best seats on this bus. So what's a good seat? Well, a good seat is a seat that ensures that as a pollen grain, you can get off at the next stop, okay, the next flower stop. A good seat is a seat that allows you to stay on that pollinator when it flies and not fall off. The other thing about a good seat is that some pollinators like to eat pollen. So bees, for example, are notorious pollen consumers. They feed that pollen to their young. And when bees get covered in pollen, they wipe it all off, they pack it onto their legs, and that pollen can't really be used for pollination. But there's certain places on bees that bees can't reach. It's like us trying to scratch our backs. Bees find it difficult to do certain things, and those little spots on bees become really, really coveted spots to put your pollen. And so the pressure mounts. And sometimes when I look into space, I imagine that this little place here on pollinators, this little sweet spot where there's so much competition, is like one of those ancient Roman arenas where these pollen grain gladiators arrive in their little teams from different flowers, and they arrive with their coats of armor and their swords, and they have these epic battles, duking it out on this little pollinator to see who's going to be the top dog, to see who's going to get the best seats on this bus. And so here you can see one of these pollen grain buses arriving, full of rival pollen grains, and now we ask ourselves, okay, as a flower, what's a good strategy here? How do you win this battle? How do you overcome these rival pollen grains? And we came up with numerous different strategies. And one of the things we thought was, well, one way that you could do it is maybe by sheer numbers. Instead of just a single pollen grain arriving, you send a whole army out. And these purple grains are the grains from this flower, so it's a whole army of grains. And we envisaged that these grains could effectively smother the pre-existing grains and trample them down to the floor of the bus where they couldn't get up at the next stop. And we imagined that if you see a pollinator with pollen grains like this, the pollen grains may be placed like a layer cake on the pollinator, where pre-existing grains might be found down below, and the new grains would be placed on top. It would smother these grains below, and the last grains on would be the first grains off at the next stop. So that might be an interesting strategy to try and test. And we thought, wow, maybe we can test it in this flower over here called Morea lurida. And I think this should be the official Stellenbosch flower, by the way. Look at this amazing color match between. But maybe when you hear what the pollinator is, you might decide not to. So they're pollinated by flies. And these flies are after little sugar rewards down here, little sugar crystals. And to get those sugar crystals, they have to creep through this narrow space. And when they do that, the anthers of the flowers smear a very thick layer of pollen onto the backs of these flies. So there you see it, and we thought, wow, this must be a great system to look at this idea of pollen smothering. It must be working here. So with the help of a postdoc, Monica Moyer, who's sitting with us today, we looked to see if we could find evidence for pollen smothering. And I'm just going to show you a little slide here. This is the back of a fly covered in pollen. You can see two colors of pollen here. You can see orange pollen. Now, that's the pollen of pre-existing flowers that the fly had visited. And the flies arrived to our flowers with that orange pollen on them. We then presented those flowers with, flies with flowers that had labeled pollen, and in this case the pollen was labeled yellow, and those flowers placed the pollen onto those flies, and what you can see here is the yellow grains lying on top of the orange grains, right? So there seems to be some evidence, at least, for this layer cake effect that we thought might be there, and this is the first time it's ever been visualized. What the consequences of this layer cake effect are, we've still got to get there. But one of the things we wanted to look for was smothering. And I'm not going to go into great depths of this graph, but I'm going to just explain to you what it says. Really what this graph is saying is that the more pollen a fly has on it, the lower the probabilities of a flower placing more pollen on there, and the fewer grains that flower is going to place on that pollinator. That doesn't sound great for smothering, right? It turns out that it's actually hard to smother pollen if it's already forming a layer on those insects. Essentially, if those seats are full, you can't oust the occupants by smothering. Maybe it's because pollen grains don't stick to each other nicely. Maybe it's because pollen is better placed on the insect rather than on top of other pollen grains. But really, it turned our theory on its head. And we started thinking, well, maybe it's not so important to smother pollen. Maybe the most important thing is to get your pollen there first in a thick layer and stop everyone else's pollen from getting on there. And that started making us think about new strategies. And we thought, well, okay, if flies arrive with all this pollen and you can't get that pollen off by smothering it, what other strategies could plants use to get their pollen onto those seeds? And so we thought of numerous different strategies, but the one that I really like, because it's quite visual, is the one that I'm going to show you here. And it's in lobelia flowers, very common little blue flowers that you'll see pretty much everywhere at this time of year. And these flowers are interesting because they have this little pollen applicator, if you can see this. It's a long tube, and the pollen is extruded through a tiny little hole at the end of that tube. And on the end of that tube are these little white bristles, little hairs. And we thought, hang on a second, what are those hairs for? And we think that those hairs are there to brush the pre-existing pollen off insects before these pollinators place their own pollen on the insects. And we haven't actually collected any data on it yet. I'm going to try and get this thing to slow. There we go. We haven't actually collected any data on it yet, but I think the best evidence here is going to be visual. You can see this pollinator visiting the flower. You can see the applicator sliding up the pollinator's head. From the other angle, you'll be able to see these white brushes brushing the pollen grains ahead of them. So from this angle here, you can see this amazing groove in the bee's head with all this pollen, and you can see these little white bristles here pushing all that yellow pollen in front of them. If you look at the next little sequence, you can even see the pollen grains flying off into the air as they're flicked off by these brushes. And as the bee retracts its head, the pollen from this flower is placed on the bee's head again. So you may have heard bizarre sounding tales of cute little insects like dragonflies and damselflies, which have these crazy structures on their penises used to extract the sperm of rival males from the tracts of the females that they're mating with. And here you can see some of those. These are different dragonfly species and their bizarre structures on their penises. Some people even say that human penises may have similar functions. But we think that we found an analogous structure in plants, where plants are able to remove the pollen from the pollinators before they're able to place their own there. So I'm going to now wind down, and at this stage, I'm hoping that some of you will be thinking, oh, plant sex is interesting. Many of you will be saying, yeah, it's interesting, but so what? What's the big picture here? And we had our honors oral exams recently, and I asked the students, so is there any place in today's society for this kind of blue sky research, research without any apparent practical application? And I was horrified when many of my students said, no, there's not. And so, much against my will, I frequently find myself having to justify the work that I do, not in terms of the richness that it brings our lives, but in terms of the material gains that it brings us. And so, if I had to indulge the pragmatists among us, I'd have to talk a little bit about the work we're doing on blueberries. And what's interesting about blueberries is that the yields of blueberries are often limited by how much and the quality of pollen reaching the stigmas. And so I have two students who are with me here today who've been working on this and using some of the ideas of these pollen journeys and the blockages and the different obstacles. They've been trying to identify the obstacles that prevent pollen from getting to those stigmas in more numbers and in better quality. And they've been trying to work out how to unblock those obstacles. And as a result, I'm hoping eventually we'll get more blueberries on our plates. I'd like to end off by thanking, I'd like to thank my parents actually for putting up with me as a kid and all the creepy creatures in my bedroom. I'd like to thank some of my mentors through my university careers, Steve Johnson, Jeremy Midgley, William Bond, and I'd like to thank them for bringing me over to the plant side. I'd like to give a really special thanks to my students who have kept my lab alive, who've kept me full of ideas, who've collected fantastic data over the years. And I'd like to thank my colleagues for a lot of fun in the field. And I would like to thank my partner, Lee, who I actually met in the field, and there are two versions of that meeting, my version and her version, and they differ slightly. And I'd really like to thank my son, Tao, who is not here with us tonight because it's going to be too late. And I'd like to thank him for showing me new things every day. Lastly, I would like to pay a special tribute to my mom. I think she would have been especially proud of me today. She really loved flowers. She was a keen gardener, she was a florist, and while I was interested in the creepy crawly things, my love for plants only grew much, much later, so I think she would have been quite happy with this. So this was one of my mom's favorite poems, Daffodils. And I've copied out the first and the last verses of this poem for you to read. But the reason why I put this poem up here is that I think the words of this poem are a beautiful example of the extraordinary effects that flowers, and remember flowers are really just these collections of reproductive parts, the extraordinary effects that flowers can have on people and on foxes too. And so I hope that I've given you some reason to gaze out into space every now and then, or at the very least, I hope that I've given you a reason to stop and smell the flowers.
Good evening once again, Professor Anderson, the Anderson family and friends, Lee, colleagues, senior leadership in the Faculty of Science, ladies and gentlemen. It's a pleasure and honor to be with you tonight to share in this rite of passage in academia. Thank you very much, Bruce, for the incredible journey that you have shared with us. Not just on that which pollen takes, but also about your research journey in this fascinating field. and to one of the most incredible 6 education classes that I have had. It's very different to the one I got in high school, I have to say. Now, one of the indicators of a really good inaugural lecture is the ability of the person professing to share deep and complex disciplinary knowledge in a manner that is both understandable to the general audience, but also clearly articulates the novelty and the complexity to the disciplinary experts. And I have to say that you have done this incredibly well tonight. In taking us on the journey of pollen, you shared concepts of polymorphism and the role that it plays in pollination. And I'm a chemical engineer by training, and so it takes me back to my days in chemistry. And it's fascinating that this concept, which I also studied in chemistry, plays out in multiple disciplines in nature. You know, in chemistry you talk about the stereoisomers and the stereochemistry. It is also fascinating to hear quantum dots, which are usually used for commercial purposes in tracking. And I think if you have cars, you would see that there's quantum dots in your paint, so you can track your vehicles if they get stolen, which is something common in this country. But it's also being used now in work, in novel ways, in tracking pollen, and to get a better understanding of the incredible journey of pollen and the pollination process. So along this journey, you introduced us to the pollen wars, and I like that kind of analogy that you drew over there, both the ability to smother and to eliminate. And once again, congratulations on your inaugural lecture, and thank you for sharing your insights on this fascinating topic. Pollen and the process of pollination plays out in our gardens, it plays out in our parks and in our various floral ecosystems. But very few of us have thought to fully understand and document this incredibly complex and fascinating nature's process. Your research brings incredible insights into this process of nature. Your knowledge and better understanding of the pollination processes shows incredible promise for not just fruit production, but also areas of plant breeding and optimizing plant productivity and resilience. Once again, congratulations on your work, and we look forward to greater fruits of your research. Well done, Bruce.
Good evening, everyone. I think I was a bit surprised when I heard that Bruce wanted to be an entomologist. And that when he got to UCT, he must have met his lecturers there that changed his mind and sent him a bit of a curveball, and he found plants quite interesting and sexy. And I think I'm a bit sad now, because I went to Wits and I didn't have that same luck that I had lecturers that really made me interested in plants. I basically avoided them like the plague my entire career. And now, having sat through this, I now see the value, and I can imagine that this is a fascinating and very sexy system to be working in. I think Bruce has been very lucky in that he's now been able to basically satisfy both of his interests, being able to do pollination stuff, where we can do his plant stuff, and actually look at the pollinators. And having now incorporated his nanotechnology, it really has opened very exciting avenues of research. And with him and his students, it's not unusual to see things on the web page that they have got some new exciting study. So they're often on our webpage, on the science webpage, for the exciting research. And I think there's going to be much more coming from him in this, with this nanotechnology. Bruce has successfully trained a large number of students and postgraduate students in the department, and has also coordinated an honours program in the last 2 years that has been through which has been a very tough task, especially now that it was during a pandemic. And he has done so very successfully. What I think I really appreciate about Bruce is that he doesn't hold back when he has something to say. He is very honest, and he has strong convictions, and he will let you know when he thinks things should be changed. And I think we need that, and he's open about it, and I appreciate that. He is a very passionate teacher, and his students really enjoy and love his teaching, and he always makes an effort in his teaching. So he will never shirk responsibilities in that he will make sure he takes his students out in the field. He will show them practically what is happening in the field. And I think he really follows the maxim, doing is learning. If Bruce is not taking wonderful photos, if you should go and look at his webpage, some of those photos are really very nice. He is chasing pollinators, maybe with a net. And one of his hobbies, of course, is photography. You can be sure he might be in the surf, where he's probably trying to catch a wave or two, and spending time with Lee and Tao. I'd like to thank you for your valuable contributions to the department, and we actually look forward to the next phase of your career and the impact that you are going to bring to future students, and that which you will bring to the department going forward. I would also like to thank Jonathan Blankenberg from the Registrar's Office, and Amira Brown, Justin Alberts, and Olivia Adams from Corporate Communications and Marketing for actually arranging this event and making it all possible. I know it's not been easy. And I'd like to invite everybody here for refreshments downstairs. Please stay behind and share in Bruce's success and in celebrating with him. Thank you, everyone.